And he's responsible for translating evidence-based behavioral interventions for neurological rehab from research to clinical care. He has a PhD from Imperial College and was a postdoc at University College London and in Western Ontario and at Johns Hopkins. Naveed's research focuses on understanding the neural substrates that control upper limb movements and how these substrates reorganize following injury, and how evidence-based software-driven interventions can be used to maximize recovery of neurological function. And I think it's really timely to have Naveed talking to us with the huge change we've seen here and around the world with telehealth. It's making a huge difference in the world. It's fantastic timing. And from what I've seen of MindMaze, it's quite impressive. So it's really great to share that with you. And thank you, Naveed, for talking to us this afternoon.
No, thank you so much, Alistair. And thank you everyone for having me. So share some slides. One of the questions I had was, how would you guys like to do this? Do you want questions to be in the middle of talks, at the end? I'm perfectly comfortable with either.
I think interactivity is always the best. So if you're happy with being interactive, that is fantastic. It'd be great.
Please, I would love this to be interactive. I don't think I'd be using my full 40 minutes, but any questions in the middle, please jump in. Don't feel shy. I will try to manage through the idiosyncrasies of trying to do this over video, but please jump in if you have any questions. It's a pleasure to be here, and on behalf of the team from MindMaze, thank you all for having us. Thanks for the introduction. I'm primarily a neuroscientist, and I've been working on upper limb function, particularly trying to understand how that works in health and how neural circuits that underlie control are impaired post-stroke and how they recover over time. And just by way of introduction, MindMaze is a startup that was founded around 2011, 2012, with a mission to try to maximize recovery of brain function following neurological disease. We've so far, through this talk, I'll hopefully try to give you a sense of the approach that MindMaze has and what we've been trying to do to translate evidence-based therapies into real-world clinical practice. And so to try to do this, I'm going to use sort of upper limb and the hand as the window into talking about the neural substrate of control, how that is impaired post-stroke. And so that window is used to also show the platform that we're trying to build. But at the end, I'll try to expand upon how that moves just beyond upper limb and expands into lower limb and other domains and other diseases as well. And so really, I'd like to talk about three things here. Using the hand and using stroke as a model. I'd first like to start off with the scientific part, which is providing you with a natural history of recovery of hand function, and primarily in the year after a stroke. And from there on in, I'll pose a problem, which is that there seems to be converging understanding of the nature of deficits that appear post-stroke and how that relates to the organization of the neural substrate. From there, hopefully it should be clear that there is a target for upper limb function that is left underserved by our current approaches to brain repair. And therein, I'll start off with just providing you with an overview of MindMaze's therapeutic approach and our platform and what we're trying to build. And then finally try to close it back into taking those two pieces of the problem statement from what we've learned from stroke recovery, our approach, and see how we're actually trying to translate sort of this missing ingredient of upper limb rehabilitation to clinical care. So to start off with, I think I'd like to talk about the natural history of upper limb recovery. And I'll primarily be using data from a study that we conducted about a few years ago, This was a multi-site investigation of upper limb recovery along 3 different sites. This was Unispital Zurich, Johns Hopkins, and Columbia University. We essentially recruited 24 patients with unilateral stroke and 14 age-matched healthy controls. And each patient and healthy control were measured at 5 different points over the course of a year. And these roughly translate into week 2 after stroke, week 4, 12, 3 months, 6 months, and about a year. So we followed them over the course of a year at different time points, sampling them particularly during the early phase where we see transient recovery, but then also in the chronic stage by 6 months and 6 months and a year. And this is just to give you a sense of the distribution initially when patients were recruited. So we see that you have a nice spread, both of patients who are, and here, of course, you're looking at the Fugl-Meyer scale, where 66 is maximal and 0 is minimal. And so you get a good distribution of patients where some patients are very mild, but then you also have a subset of patients that are very severe. And you sort of see a bimodal distribution with a few missing patients in the middle of the distribution. If we now look at the Fugl-Meyer as it recovers over time, you see what you would expect, which is if you look at the non-paretic side, because these are unilateral stroke patients, they of course have 66 throughout, meaning that there appear on the Fugl-Meyer scale, there's really no impairment that's captured. But on the paretic side, of course, you see that in the initial phase, the Fugl-Meyer score is low, and then it recovers slowly over time, but it never really fully recovers back to baseline, which in comparison to the healthy side. And then, of course, most of the recovery is within the early phase, which is within the first 3 months. And this is just to show you the similar plot, but with a measure for strength of the hand. And here you see much more fine-grained breakdown of how patients are impaired in the hand. So in blue, you see healthy controls. MVC is the maximum voluntary contraction for the whole hand. And so with healthy controls, you basically see there are about 20 newtons on average. On the non-paretic side, you start seeing, in comparison to the Fugl-Meyer, you start seeing that there's an initial dip, but then, I mean, that's negligible, but then it slowly recovers to baseline, and that's all within the first 3 months. But then now, in comparison, you see a cleaner curve where the paretic hand has lost strength, and again, it recovers within the first 3 months, and then it recovers. But then, and it basically asymptotes while you reach 6 months and about a year. Any questions so far? Okay, great. Now, basically the idea was that in addition to these parameters for Fugl-Meyer and strength, what we basically wanted to understand was, could we track the recovery of fine finger control over the course of the year? And so in doing so, we had a couple of challenges. The first was, of course, we needed to build sensitive assays for measuring isometric forces produced at the fingertips. But we also needed to produce a task and a measure that had to have a couple of properties. The first was that it needed to be tailored to a patient's individual impairment. So if a patient had low force, then the task need to be sort of tailored to the ability of a patient to do force with one finger. But then on the other side, the metric also needed to be sort of force agnostic, because you wanted, as we saw, patients have deficits in strength, which recover over time. So if you wanted to measure which allowed a comparison between patients as they recovered over time or patients compared to controls, you wanted that measure to reflect dexterity, you need to have a measure which sort of takes the strength contribution of the patient out of the equation so that you can actually compare a measure which speaks to dexterity itself, but it's not confounded with the fact that strength changes over time. Okay. So what we did was we built these sorts of piano-like devices where your hand fits into an ergonomic keyboard. Your hand is strapped in, your wrist is strapped in, and underneath each finger is a force transducer, which is quite sensitive. So when you press, the device measures a force, and the force is then translated into this sort of video game that you see on screen, where you see all 10 fingers with the white line showing the force on that individual finger. And essentially the task is that you're shown a force or a target force on the screen. And what you have to do is press with the finger in order to reach the force while keeping all of the fingers stable. And then the first phase of this, of course, is that we measure the maximum force being produced by each finger on both hands. And then we test at different force levels, which would be 20%, 40%, 60%, and 80% of that force. So you're always within the operating range of that finger. And so when you do this, this sort of task has been studied quite a bit by Mark Latache and colleagues. And when you do, of course, you see a nice force trace where, for example, you over time, the instructed finger produces the amount of force that is required. But when you do, you also see pooled averages for all the other fingers in the hand. So in this case, if let's say you were pressing with the index finger, and the task was to only press with the index finger, you see co-contracted forces on the other non-instructed fingers of the same hand. And this is a phenomenon that's been sort of characterized by Mark Latache and colleagues and others, and it's a phenomenon called enslaving. And the idea is that it's very tough, given the musculature and neural control and circuitry of the hand, to make isometric contractions of just one finger. And there's sort of a spillover into adjacent fingers of the same hand, which lead to these sorts of enslaved forces. And so these enslaved forces are seen in health, and they're minimal compared to the force produced by the instructed vision. Now, on a related note, but something that is studied not related to enslaving, which is equally important, which is that if you were to ask someone to press with just one finger on one hand, and now not only measure the forces on the uninstructed finger of the same hand, but started looking at the forces produced on the other hand where it was not instructed to move, you start seeing force produced on the uninstructed finger of the uninstructed hand. And these forces are essentially called mirror movements. And again, mirror movements have been shown in healthy controls. And the idea here is that we're going to try to characterize both enslaving on the instructed hand and mirror movements on the uninstructed hand, and try to construct a picture of how this evolves over time and what that can tell us about the neural circuitry. And so essentially the task is that we first ask patients and healthy controls to first press as hard as you can with an individual finger, then produce forces at about 20%, 40%, 60%, 80% of the maximum voluntary contraction. And so what you see is, of course, that if you ask them to press down with their fourth finger, when you're at 20% of your max, you don't really see enslaving in the other fingers. But as you start ratcheting up the force requirement on an individual finger, the amount of enslaving on uninstructed fingers also goes up. And what you can kind of do now is sort of plot this, where you plot the active force on the instructed finger against the RMS force on all the uninstructed fingers of the same hand. And when you do so, because we're measuring at 4 different force levels, which is 20%, 40, 60, and 80, you start seeing a relationship emerge where as the active force increases, the overall amount of passive enslaving force also increases. And to a large degree, this can be characterized by sort of a linear relationship. Now, of course, this is an example of one person where they had significant enslaving in the hand. Here now you're seeing another patient where you're asked to again produce with 20% and 80%, but this time you can notice that even though the forces produced are smaller than the first patient, there is much less enslaving in the other fingers of the hand. And so again, you can plot this on the same plot. And really, I think the point here is that these curves can sort of be approximated by the line of best fit, which fits all these data points. And the slope essentially tells you per unit applied force, what is the amount of enslaved force in the other fingers? So in a way, the slope is, for example, so in this case, if you have a high slope, that basically tells you that you have sort of quote unquote poor control. But if you have a low force, that basically tells you that you have good control, because per unit force you're applying, you're producing less enslaved force. And so this slope by itself is what we call a strength invariant measure of dexterity, where dexterity is this sort of finger individuation task where you're only instructed to produce force with one finger while keeping the forces on the other finger stable. And this is the reason why we're particularly interested in this task is because, of course, post-stroke, what you see with patients is the production is a phenomenon called muscle synergies, which is not the mathematical description of it, but the clinical description, where patients, when they're asked to produce isometric forces or individual forces with one finger, you see a much greater spillover of muscle activation on other fingers, meaning they lose the ability to make like fractionated control of isolated joints. And so this measure is a way of capturing the amount of spillover of muscle synergies into the other fingers of the hand during this fine finger individuation task. So moving forward, what we'll be basically doing is we're of course characterizing all these different forces. We're going to measure patients at different time points and then look at the evolution of this slope over time, both in the instructed hand and in the uninstructed hand. So you basically now see two panels. The first panel is basically showing you the passive forces on the enslaved forces on the paretic hand and measured over 5 different time points, 2 weeks, 4 weeks, 12, 24, 52. And on the right side, you're basically seeing the mirrored forces on the non-paretic hand. And I want to stress that in this sort of paradigm, it's always the paretic hand that is moving. Right? So the first thing that you see, and then on the y-axis, of course, you're basically plotting the slope. And this is plot as that per Newton, this is the force, this is the amount of enslaving or mirrored force per 1 unit of force applied by the instructed finger on the paretic side. And so what you see here basically are healthy controls on both sides, where of course healthy controls basically show that per 1 unit applied force, you get about 0.05 newtons on the paretic fingers, on the fingers of the paretic hand. But then you also see mirrored forces as we would in healthy controls, but those mirrored forces are an order of magnitude smaller than the enslaved forces which are produced, which is of course what we know. Mirrored forces are much smaller. Now, if we start looking at enslaved forces and their evolution over time, what we see are a couple of features where, as for strength, it's in the immediate aftermath after stroke, enslaved forces are higher, which would be the emergence of synergies. Patients have reduced control, which of course resolves over time, but it never really resolves fully back to baseline in comparison to healthy controls. Most of the recovery is within the first 3 weeks, as shown for strength and as shown for the Fuggenberg. Okay. And this is basically just to show you statistics that it recovers over time. Now, in comparison to that, what you see is a sharp increase in mirroring immediately after stroke. Again, as for enslaving, it recovers over time. But in comparison to enslaving, it does sort of resolve back to baseline, where now it's statistically insignificant from healthy controls, right? But again, as for enslaving, most of the recovery is within the first 3 months post-stroke. So basically what you see here is that, and there's a particular reason why I'm showing both these plots on the same axis as opposed to looking at enslaving first and then mirroring first. It's basically to demonstrate that as a singular event, stroke causes the emergence of 2 phenomena, one in the paretic hand itself, which is a loss of control, and there's sort of an unmasking of a similar phenomena in the non-paretic hand, both of which are immediately exaggerated post-stroke. They resolve over time, and they resolve along the same time course, which is within the first 3 months. And there's a massive change in the recovery of those parameters in the first 3 months. Now, even though there is significant change within the first 3 months, one of the ideas in the field was that because fine finger control is primarily mediated by corticospinal pathways from the motor cortex down to the spinal cord, down to the muscles of the hand, is that mirror movements and enslaving are essentially primarily corticospinal phenomena. And therefore, we should be able to see some sort of neurological correlate in the motor cortex. And so Nick Ward showed in longitudinal studies and also in cross-sectional studies that he could see signatures of increased activation in the motor cortices for in the ipsilateral motor cortex or in the contralateral motor cortex post-stroke. And there seemed to be sort of an interhemispheric imbalance that was created in the immediate aftermath after stroke as compared to healthy controls. Basically, the idea was that you have a healthy signature of activation in the motor cortices, which could be either the ipsilesional or the contralesional cortex. However, that is sort of changed as a result of stroke, and then we should be able to use imaging to be able to capture that. Now, what I'm showing you here is in fact a null result demonstrating that if you do imaging on the same patients in the same cohort, you in fact find no change in mean signal over time. And I'd like to walk you through exactly what this means. So on the x-axis, of course, is time, but on the y-axis is the beta coefficient calculated through signal intensity changes in the motor cortices, both in the primary somatosensory cortex and the primary motor cortex. In the contralateral cortex, when you move your hand, in the contralateral cortex, we know that there is an increase of activation. Simultaneously, we know that there's sort of a suppression of activation in the ipsilateral cortex during unimanual finger presses. And so that's why what you see as these dashed or these green lines are essentially the control activations in the contralateral and the ipsilateral cortex for finger presses. Now, what you see, of course, in the activation for the contralateral and ipsilateral hemisphere is that, in fact, they're completely overlaid on top of the healthy controls, meaning they're no different from healthy controls. But also what you see is that in comparison to the behavior, which showed significant longitudinal change over time, there is no longitudinal change in signal intensity for the sensory cortices and the motor cortices here. So then we get to the question, well, if there's no change in the cortices, then what can we learn more from looking at the fine-grained analysis of the behavior itself? And this is where we turn to looking at sort of two different components of control in the paretic hand itself. So previously I showed you recovery of strength, and I showed you recovery of the individuation as measured to our measure. Now, you would say, well, they both recover similarly, where the recovery is within the first 3 months. They show equivalent time course changes. But here I'm showing you a sort of different view where I'm not plotting these points as time, but each point is in fact a patient on this plot where time is basically thrown away, but you have strength on the x-axis, and then you have the individuation index on the y-axis, with the only difference being that the individuation index for clarity is switched in sign, which means that higher numbers are more recovery. And when you plot it this way, what you see are essentially a biphasic curve that emerges. Basically, and of course, each dot is essentially a patient, and the sort of ellipse that you see in blue with a dot is the healthy controls on that same axis. right? So basically, healthy controls are to the right of strength, meaning that they have significant strength, but they're also high on individuation, meaning they have high fine finger control. And of course, if you're bottom left, that means you have low strength and low control. Right? And this plot, basically what it shows you is that up to a certain point, which is a strength of 0.6, which is again normalized strength, what you see is collinear recovery between strength and fine finger function. Meaning that for every unit of strength that you recover, you also recover a unit of dexterity. But beyond 0.6, that relationship breaks, where you can essentially recover additional strength, but that doesn't necessarily correlate with increased recovery of fine finger control. So in a way, what this basically is showing is that after a certain point, strength potentially continues to recover, but fine finger control saturates. Right? So this was the situation on the paretic side. Now I'd like to take a look at the non-paretic side and what is happening there in terms of a fine-grained analysis of behavior. Now, with mirror movements, one of the ideas was that essentially, and it's in the name itself, which is that the idea is that if you produce any movement with, let's say if you start producing wrist flexion, mirroring would dictate that you should start seeing wrist flexion on the other side. Or if you press, or if you produce finger flexion of the index finger, then you should start seeing like flexion of the index finger on the other side. So basically, the name itself is the idea there is that, in fact, mirroring is primarily a homologous phenomena. It's like for like, it's muscle to muscle, right? And one of the reasons why this sort of exists is because for some reason, we haven't really looked into non-homologous muscles and fingers during any sort of task where you can actually make the distinction about how much is it homologous and how much is it heterologous, right? And so here, what you're basically seeing is just the patient at week 2 and a healthy control compared to healthy control, right? So, and of course, on the y-axis is basically the amount of the magnitude of the mirroring on individual finger pairs. And so here, basically, I'm breaking down the curve plot that you saw for mirroring, but now as compared to individual base pairs of fingers. So you have an all possible combination of active fingers against passive fingers. But again, it's also broken down into when it's on homologous fingers, so when the active and the passive are the same, versus when it's any other combination. So the first thing that strikes you, of course, is that yes, there is significant homologous mirroring, but it's not over and above mirroring on the heterologous fingers. So in fact, it's not, the point is that mirroring is not simply on homologous fingers, it's really spread across the entire hand. The second thing is that it seems to be a pretty good copy of what is happening with healthy controls, right? And it seems to just be an amplified version where whatever the mirroring pattern was on in health is just exaggerated in patients post-stroke. And we can, of course, run statistical tests on it to show that, in fact, when you compare the patient's pattern against healthy controls, you see reasonable correlations, but these correlations remain stable, meaning that the patient's pattern is statistically the same as healthy controls, and it doesn't change over time. All it happens is that it's exaggerated post-stroke, and then it recovers back over time. Now, I'll conclude all what these different pieces mean, and then I'll try to put it together about what it tells us about the neural substrate. So what have we learned? Basically that by using this measure where we can dissociate and sort of extract out the strength contribution of fine finger control, we learned that enslaving and mirrored forces appear early after stroke, and then they show longitudinal time course changes, and they recover within the first 3 months post-stroke. What we also know is that so far, given the sensitivity of the metrics and the measures we have with fMRI, it doesn't appear that these sort of longitudinal time course changes in enslaving and mirroring could be explained by signal intensity changes in the motor cortices, which sort of goes against some of the work that's been done by Nick, where Nick essentially had about like 10 patients where he measured longitudinally. But most studies that have sought to make a link between enslaving, mirroring, and motor cortical activity have in fact primarily been cross-sectional studies, not longitudinal studies. So they missed any time course changes. And of course, one of the points here is that mirroring is not limited to homologous fingers. Now, this might seem like an interesting point, but in fact, it goes even further in the direction of the fact that it cannot be explained by cortical activity, because Jorn Diedrichsen and John had this paper showing that in fact, when you produce movements of let's say the index finger, and you look at activation patterns in the contralateral hemisphere, now you're not looking at signal intensity, you're looking at activation patterns across different voxels. That pattern looks remarkably similar to whether you're pressing the index finger on the contralateral hand or the ipsilateral hand, meaning that in fact the activation patterns, if you were to predict homologous mirroring, would look exactly the same. So in fact, given that the activation patterns for movements of either finger on the hand look exactly the same in one hemisphere, it would in fact predict that mirroring should be a homologous phenomenon, where in fact, behaviorally, if you look at it, it's just spread across the entire hand. And then finally, one of the things we've learned here is that beyond a certain point, any further improvements in strength are not correlated with fine control. So, I mean, it's great. I mean, there's a lot of different things that we can learn about this, but what does it all mean? And what does it mean in terms of the substrate that controls movements and how it is altered post-stroke? Well, I mean, what we know, of course, is that like movements of the hand are, or like muscles in the hand are controlled by ventral horn neurons in the spinal cord. And these provide the primary hands as drivers of input. This was a question that Lawrence and Kuipers in their seminal study looked over very carefully. And essentially their thought process was the following, which is that if you look at primates and you look at the cross-section of the spinal cord and you look at all the inputs to muscles of the hand and where they come from in the ventral horn neuron of the spinal cord, they found that, in fact, if you trace it back up, there are multiple different motor pathways that contribute as inputs to the same pool. So their question was, well, if you got so many pathways that contribute to the same pool, how do they interact? And which one is more important than another for control? And so like all good electrophysiologists, they basically started cutting. So first they started cutting the corticospinal tract. Right? And when they cut the corticospinal tract, of course, what they found was that the arm became completely flaccid, meaning the animal couldn't move. Right? But then over time, what happened was that the animals remarkably recovered nearly fully, in the sense that they could essentially grab the cage and fully support their body weight. But what they found was that, in fact, they could not do really fine movements. They couldn't basically go into a well and reach for a pellet and then feed themselves. And so one of the conclusions that Lawrence and Kuipers drew from this first experiment was that, in fact, they said that these corticospinal pathways that run to the spinal cord are primarily responsible for fine finger control, because that's what you lose when you ablate or cut that pathway. Now, what they then did was that when the animals recovered, they started cutting other pathways. And one of the ones they cut was, of course, the rubrispinal pathway. And there, essentially, what you found was that the fine finger, like whatever residual recovery that the animal had, that was decimated. Right? And so that led them to conclude that even though the arm had recovered in the absence of the corticospinal pathway, and that the animal could retain, like even after cutting the corticospinal pathway, the animal did recover control, but just not fine finger control. And that by cutting the rubrospinal pathway, they lost any residual recovery they had. Is that, in fact, they said that while the corticospinal pathway was primarily mediating dexterity, other brainstem pathways were sort of involved in gross control of the upper limb, strength, maintaining your body weight, but then perhaps also had mild ability to also perform some dexterous movements. And so that's how they sort of split this landscape, where the newer corticospinal pathways were dexterity, and the older rubrospinal and reticulospinal pathways were sort of gross control and some level of fine level control. Right? And so what we essentially argued here is sort of the human equivalent of Lawrence and Kuiper's, where we said that you sort of, in this first phase that you see where there's colinearity between strength and individuation, it's in fact the older pathway, which is, of course, humans don't appear to have the rubrospinal pathway, but we do, of course, have the reticulospinal pathway, which is what we feel is the equivalent. And so the sort of colinearity that you see is essentially the older pathways sort of compensating for the loss of corticospinal control. And so while they're primarily involved in the recovery of gross strength, they can potentially do a little bit of dexterity. But then what you essentially lose is the corticospinal pathway, which is involved in mediating fine dexterous control. And so in this paper, we sort of argued that this is evidence for the fact that there are two systems at play, even in humans, as in primates, and that stroke sort of uncovers how these two systems are interacting with one another. And this also, this also comes back to the fact that why do we, in a way, this also explains why we showed enslaving and mirroring on the same plot, is because corticospinal pathways, as you would know, are primarily lateralized. So after the first year of birth, most of the fibers basically cross over to the other side, and any ipsilateral fibers are basically pruned away. If I have my numbers right, it's about 95%, no, 70, 85% and 15% on the, so 85% crossover, 15% are on the same side, but those get pruned over. Well, sorry. They're 50%, 50% immediately after birth, and then they get sort of 85, 15 over the first year of development. In comparison, the reticulospinal tract is more bilaterally organized. So it's 60%, 40%, meaning that if, for example, you lose corticospinal control, of course, is mediating the action of reticulospinal circuits. And if you take away the inhibition of the reticulospinal circuits and they overexpress themselves, because they're bilaterally organized, the behavioral consequence is going to be on both hands, meaning that any exaggeration of reticulospinal pathways is going to lead to spillover in the paretic side, but also spillover in the non-paretic side. And so, in fact, the model that we're arguing for is that essentially what happens after stroke is that you have the corticospinal pathway in health, you have the reticulospinal pathway in health. They're basically working together to produce fine control when needed and strength when needed. When you blow out the corticospinal pathway, the reticulospinal pathway is sort of unmasked and it tries to compensate where it can. And therefore you start seeing an exaggeration of enslaving and mirroring on both sides. But essentially what happens during a recovery is that somehow residual corticospinal pathways start to re-exert control over these reticulospinal circuits. And that's how the story sort of pieces together these disparate pieces of enslaving, mirroring, and how the system recovers. Right. So that would kind of conclude the first section. Are there any questions so far?
Thanks, Naveed. I think I was, I don't know if you mentioned this in the beginning, but that the group of people, are the strokes all similar, or is there quite a difference in the type of stroke and where the stroke is in this group?
There are significant differences. I mean, but primarily they were primarily internal capsule strokes, mostly.
Okay.
But then, of course, you did see some basal ganglia, some cortical strokes, a few parietal, but we primarily tried to recruit unilateral stroke patients. So, and even though there was some variability, we didn't have the statistical power to sort of do a more fine-grained analysis based on lesion location.
I think we've got about 10 minutes left, so I'm just wondering about how long you wanted to talk on the next section.
I mean, the next sections can be actually the next sections we can keep shorter.
Okay.
And so one of the points is that, well, okay, given this sort of model for recovery, the question then becomes, well, how do you promote further recovery of fine hand control, right? Because really what it seems like is that dexterity is sort of saturating during spontaneous recovery. Now, what I'd like to point out is that in the trial, we did really have an exaggerated recovery or rehabilitation phase, patients who were receiving standard of care. Now, so the question then becomes, well, is this the best we can do? Because this is the intrinsic limitation of the neural substrate and its ability to recover. And for a long time, that was sort of the idea, is that that's basically we're hitting ceiling. And then, but we know that that's not the case in animals, because in animals, when we give them strokes, the difference is we give them intensive therapy. Whereas in humans, for some reason, the system is designed not to provide high dose, high intensity rehabilitation, right? And so there's sort of a weird mismatch between what we do with animals and what we do with humans. And so Nick tried to come along and said, well, I'm going to try to provide a service where for my cohort of chronic patients, where I know that they have basically plateaued and they're not recovering more, I'm going to provide high dose, high intensity upper limb intervention, where in standard of care, for example, in the United States, you would get about 16 to 20 sessions as outpatient therapy post-rehabilitation. That would approximately be about like 16 to 20 hours scheduled, but really it is about 12 hours time on task on average, of which a fraction, if you look at the Catherine Lang study, where in a 45-minute session, about 10 minutes is dedicated to the upper limb. So now we're going into fractions of how much we actually deliver to the upper limb. And so Nick basically came along and said, okay, well, I'm just going to do a targeted high dose, high intensity upper limb intervention program, 90 hours of therapy spread over 2 to 3 weeks. And then chronic stroke patients where the idea was that in fact, they cannot recover significantly over baseline. He in fact showed significant increase about 1155 points. But still the problem was that what are the raw ingredients that he used? And if you speak to him and his therapist, he basically says, well, it's a lot of different factors, but the raw ingredients are basically motivating time on task, somehow through the therapist, some form of robotic or arm de-weighting of the upper limb, and then some combination of functional electrical stimulation and functional tasks, right? There was some mix of this, but the paper was unclear about what exactly that recipe was. But regardless of what the recipe was, the point here is that how do you scale this? Because Nick is essentially delivering 90 hours of therapy per patient. The wait list for his clinic is about a year. So by the time the patients get to him, they're of course chronic, they're not acute. And then the question then becomes, who pays for this as a service delivery in the medical profession? And these are non-trivial questions, which of course might not seem scientific, but they're very, very relevant in terms of service delivery. And so that's where we basically looked at the problem and said, well, I mean, if you look at rehabilitation and recovery from neurological diseases, there seems to be a lot of onus on drugs, But there is no drug for stroke recovery. There is no drug for spinal cord recovery. And so this sort of gap has been, like, it's tried to be filled by medical devices, right? And the sophistication here really is you build a big robot, it's going to be expensive, you strap the patient in, and somehow through the action of the robot, the patient will recover. But if you look at the Rattle study, which was about an 800 patient study in patients, what they showed was, in fact, there really was no statistical change in patients' recovery with sort of these robotic interventions. And so what we're basically going back to is back to basics, which is that if you really want to affect change in these patients, we have to basically provide behavioral intervention. And these behavioral interventions need to be high dose, high intensity, and they need to be scalable. And the approach that basically what we're taking is that we deliver behavioral interventions through software. And this is sort of this new field of digital therapeutics, and we're trying to establish the field of digital therapeutics in neurorehabilitation. And the way that we work is that, of course, if you want to provide behavioral interventions, you need to be able to sensitively measure behavior. And so we develop our own hardware, we develop our own algorithms to sensitively assay behavior and use that as a control signal into purpose-built video games, where the video games, of course, have a specific therapeutic target in mind. And we essentially can develop, we have games for upper limb, lower limb, gait, balance, cognition. And essentially, if you use a combination of a hardware and a particular software, you can make a purpose-built game for gait and balance or upper limb. But what you can also do is use combinations of hardware, algorithms, and games to build the next generation of kinematic assessments on top of the Fugl-Meyer and the ERA. Right? And both are equally relevant. And so the platform that we're building in MindMaze is essentially a sort of a software-based continuum of care service where the patient is managed all the way from when they come into the clinic to using the system at home and anything in between. And it basically is this system of like a modular components where you have software which provides the games, and then you have peripherals and algorithms for full body motion tracking, peripherals for hand and finger training, an exoskeleton for 3D arm de-weighting, functional electrical stimulation and tasks, and then of course the accompanying assessments that go along with it. In the acute, subacute, and chronic phase, we're going for high dose, high intensity interventions. And then at home, one of the things we're trying to work on is that we've submitted a code for, in the United States, for asynchronous rehabilitation. And so when the device goes at the patient's home, with the codes in the United States post-COVID, essentially these patients can use, therapists can in fact use the modifier to provide synchronous telerehabilitation, but there is a door available, opportunity available, where through the system, the therapist can set the patient homework, and the patient can continue to do the homework to increase on, multiply upon the dose, which solves Nick's problem, because this then becomes scalable. And what we're working with CMS and the AMA is that we've asked for a code for asynchronous rehabilitation. I can show you a very quick video of how this works.
MindMotion Go is a therapy tool designed to motivate and engage patients' participation in active therapy and to help increase their rehabilitation dose. Through gamified tasks in virtual environments, patients are motivated and perform several repetitions of movements. These fun and engaging activities encourage patients to play longer. MindMotion Go is intuitive and easy to set up. The system is ready to use in 5 minutes. MindMotion Go has a collection of engaging, gamified activities with 17 different therapeutic movements. Therapists can customize training sessions and calibrate each exercise based on the patient's needs. The level of difficulty can be adjusted to an individual patient's abilities and progressed as the patient improves. While exercising with MindMotion Go, patients receive audio and visual feedback, as well as real-time compensatory detection. This feedback encourages proper movement patterns and may lead to more independent practice. MindMotion Go allows training in standing and sitting. MindMotion Go simplified activity reports allow therapists and patients to track improvement in scores and progress goals together. Discover how MindMotion Go can help motivate patients to reach their full recovery potential.
And I'll just leave you with a couple of thoughts, which is that I'm showing you data from about 200 patients in the immediate aftermath of COVID, which is between Q1 and Q2. And what you see is a substantial increase in usage of the device, but you can see that the device is being driven by telehealth. But what's interesting is that telehealth is not really coming at the expense of synchronous sessions, but in fact, the future is probably going to be some sort of hybrid between telehealth and synchronous, where like a mix of what a clinic can deliver depending on their capacity is probably going to be the future. And finally, just to close the loop between like the sort of prototype work we did in the upper limb and how we're delivering this sort of care with our new systems, is that this is a new peripheral we designed, which of course can't do individual finger control, but it is a highly sensitive measure of force produced at the hand. And so this is an example of an assessment that we are building for grasp dexterity, where again, the concept is the same, as rather than making individual finger movements, you essentially test grasp at different force levels, and then you subtract out the RMS score on a curve following task. And similarly, of course, these games can be used for like sort of pronation, supination, grasp control, so that you can use different combinations, because the sensor is not just grasp, but also pronation, supination. And that's it. This is just to show you the clinical trials that we're running in order to do evidence generation for reimbursements and for our evidence generation pipeline.
Naveed, may I ask you a question? This is Amit from Brisbane. Sorry, I missed the first 23 minutes of your thing because of a link problem. So
no worries.
If the question sounds a bit daft, you know why. I come from a sort of a personal thing, because my surgeon mother had 9 strokes before she passed, but each one had a different implication on her. And for the first 5 or 6, she sort of recovered quite a lot, except her vision, which was impacted. Now, if I focus on one aspect of that in one stroke, she lost her speech more than in others, and it came back through practice. So I'm just trying to join the dots up here is that what you're trying to say through this is you can actually, through various sorts of exercises, and yours is a sophisticated way of exercising the arm, you've proved that you can pretty much come back to where you are. Is that the gist of what you're...
I can try to summarize the gist of it. So the gist here is that from animal literature and human natural history studies, we now have an understanding of, let's say, one of the key deficits post-stroke, which is upper limb hemiparesis. Why that occurs, the neural substrate, and what we think is going on. Using that as an example, like 70% of patients post-stroke would have hemiparesis. Using that as an example, we now, of course, know is that there are behavioral interventions which, given at the appropriate dose and intensity, can serve to, if not bring you, of course, they won't bring you back to baseline, but they can have the potential to give significant recovery, which a few years earlier, we didn't even think that was possible. Now, the problem then becomes is that's where the science stops, and then the practicalities begin. Well, how do you provide that sort of high intensity rehabilitation to patients outside of the current organization of the healthcare systems. And that's where these software-based interventions come in, which says that, okay, if you want to train upper limb dysfunction, here is a package of peripherals and software, which you can use across the continuum of care from acute to home, with synchronous with a therapist or asynchronous through a device. Now, you make an interesting point, is that not all patients would have similar deficits given the nature of their stroke. But the idea is that the platform is just a conduit through which different evidence-based interventions can be provided. I just use upper limb hemiparesis as the first example of how we're doing it. Of course, in the pipeline are gait, balance, cognition, and the rest, which of course is delivered through the same platform.
But what actually happens in the stroke is when you get the stroke, it impacts your, say, if you're focusing on the arm, so it'll impact your arm, right? When that clot dissolves, what's actually happened is that the muscle in your arm has got weak. And all you're doing is you're teaching it to get back to normal. Isn't that what it's all about?
I would say it's a little bit more sophisticated than that, in the sense that the brain evolved over millions of years with competing different pathways contributing to movement. And these pathways interact in very different ways to produce movement. And what happens post-stroke is that depending on the location and the nature of the deficit, the sort of behavioral deficits that are produced need to first understand what the interplay between the movement generation pathways are before they can sort of recommend the appropriate therapy. And the evidence that I showed earlier was that traditionally in rehabilitation of the upper limb, dexterous control of the upper limb has really been sort of underlooked as a therapeutic target. And so I showed data, in fact, showing that really it's one of the key targets that we need to be able to intervene on. But in order to intervene on that as a target, you need to be able to sensitively measure it and intervene on it with the most appropriate hardware and software. And that's so far been missing. And that was sort of the gist of the first half. It's
a great question, and thanks for that. I think it underlies that stroke's different for everybody. Every stroke is different, and even for the same person, each stroke could be different. But everyone likes to play games, and at the moment, we love to interact with our digital devices more and more. So it looks like a great way forward. I was wondering if there are questions from others in the little time.
How can we get in touch with Naveed after this?
I'll drop my email.
Thanks, Louise.
Okay.
Yeah.
So I'm a stroke researcher and trained as a physiotherapist. And yes, I obviously totally agree that we know and have known for some time that the only way patients are going to get better, there's no magic bullet. They have to do lots of repetitive task practice. What frustrates the hell out of me is how come everyone, it's obvious that we should use, software and this sort of technology and games to do this, because we're in that sort of world now. So why wouldn't we? But everyone all around the world is inventing these. Why does everyone have to invent another different one? There's been some come out of New Zealand that we've been using, the Able X and the Able M, for years. And I'm not saying that yours isn't better than that. I'm just saying, why can't everyone get together and just design a perfect one? Why is it like mobile phones? We have to have millions of different sorts and none of them work the best that they could. It's very frustrating.
I completely hear where you're coming from. I think, if I may, I think the problem has been that it's primarily been driven by, these companies have been driven by engineers, where the idea has been, let's make a game and then let's figure out everything else that comes afterwards.
And who can use this game then? Let's make a game and then we'll figure out who can use it.
Exactly, who can use it. And they design the games for healthy, and then of course, there are all sorts of problems. But really the challenge is, of course, evidence. I mean, the difference here, and I'm not trying to make a distinction between us and them. Of course, what will happen, as with any industry, is that there's a growth phase and then there's a maturation phase where there's consolidation into a few platforms, and that will happen. But the larger point here is that there are two things that we as a company focus very heavily on. The first is evidence, because it's one thing to build a game, it's another thing to build a game which is gone through a sequence of clinical trials to demonstrate efficacy. Those are two fundamentally different things. The second thing is that we're putting as much effort into understanding reimbursement pathways and building new codes for these games as we're doing in developing the games themselves. And that second path, I think, is one of the reasons why most of these companies falter, because they feel that just build the game, give it to the therapist, and somehow they will figure out how to maximize the dose within the constraints of the healthcare system. That's primarily been how they've been operating so far.
Do you know why I think they falter? They're
Please.
too expensive. There's no money in the health sector.
And that's the point.
You have to realize there's no money in the health sector. And so unless you make it, no matter how sophisticated and even how much evidence you have, if it's not simple and cheap enough, it won't get taken up. It won't get used.
Or,
unfortunately.
Or yes, that's one. Or the other thing is that you work with regulators and payers to get new codes for these software interventions. And so in 2 days, we will be defending our asynchronous code in front of the AMA in the United States. So fingers crossed. So the point here is that yes, there's science and yes, there's evidence, but I completely take your point that there's equal amount of effort in the coding and how we pay for it. Because if we don't figure that out, then all of this is kind of, it's the same as what came before.
Yeah.
I think it's a lot to do with getting comfortable with the various programs. And if you're in front of the right person and they can see the value, then they'll take Naveed's. And if the New Zealand guy comes along, that's fine. But they've already got Naveed established in the system. So unless there's a significant delta there, you're not going to change. I think that's how it works. First mover advantage.
I mean, we're honestly not even the first mover to be. We're a big player, but we're certainly not the first. But we put our markers on evidence and trying to understand who pays for it, because that's really what will crack it. And they will be consolidated.
Any stroke, you ask any stroke victim, what do they want to do? They want to get back to normal. First, they have a depression that they've got it. They get over that and they want to get back to normal. So I think there's a big space.
I mean, full disclosure, I have a stroke myself. That's one of the reasons why I'm in the field. But yeah, I mean, and again, the other thing also is that there's an underestimation on clinical trials and actual implementation. If you look at clinical trials and sort of medical devices that have been used, they all claim to be like adherence rates for like 6-week programs to be 98%. And then you ask them what happened after the trial ended, and adherence goes down to 40%. That is a significant challenge when you're in actual clinical implementation versus a clinical trial. And that's another thing that we pay close attention to.
Hi, Naveed. May I ask you a question?
Please.
Yeah. In the first part, you mentioned like the enslaving and the mirroring cannot be explained by signal change in the motor cortex. So does that mean like after stroke, the motor cortex activity is remained the same as the healthy people, or it because the stroke not impaired the cortex of that part, or because the enslaving or mirroring is a passive like activity? So
that's a very interesting question. To the extent that our 3 Tesla scanners can measure appropriate signal intensity changes and appropriate measures, what I can tell you is that plot is the highest N plot that I know of on longitudinal data in stroke, showing that there is no difference between healthy controls and patients. That I can say, that much I can say unequivocally, right? And then it's a question of, well, is fMRI really the right, is there really true changes which are at a much smaller resolution that fMRI can't pick up? That's one question. That's one possibility. The other possibility is that, in fact, most of the reorganization might not be cortical, might in fact be a mixture of cortical and subcortical, which goes in the direction of the Stuart Baker and Zaini work, where they in fact showed that there's a lot of strength changes in the reticulospinal system, which we are just not sensitive to with fMRI right now. So it's somewhere in between the two is probably the right answer. And my guess about why older papers have been showing signal changes is probably because I think as a field we've been there are a lot of tissue changes that happen post-stroke. Dysthesis is going to change signal intensity. So it's tricky, but to the extent that our measures are sensitive enough right now, yes, signal intensity for healthy controls and patients are the same post-stroke.
Okay, thanks. This is great for use brain-computer interface to detect the movement if the cortic activity is similar for healthy and stroke victims. Yeah.
I mean, they're not BMI, but of course there's the competing model that we are arguing against is sort of the interhemispheric imbalance model, right? Which is that post-stroke, one hemisphere sort of either helps compensate or sort of gets in the way of the other. What I didn't talk about was, as part of the data set, we also collected TMS measures for intercortical inhibition. And what we found was that, in fact, this sort of notion that there is excess of cortical inhibition from one hemisphere to the other is in fact epiphenomenal. And I can share the reference with you. What it in fact shows is that it is, I mean, you do find an imbalance in interhemispheric inhibition, but it emerges after 6 months. But most of the behavioral recovery is within the first 3 months. And within the first 3 months, interhemispheric inhibition is the same between controls and patients. What I also didn't show is fMRI data, which is a paper that's currently in review. Is resting state data, because of course, fMRI is corrupted by behavioral confounds. Resting state to that extent isn't. And analyzing resting state in the same cohort of patients, we in fact found no difference between patients and controls. So all signs point towards the fact that whatever measure that we're using for cortical activation, inhibition, they seem to show no change, yet behavior does. So maybe we're just not looking in the wrong place, or our tools are not sophisticated enough.
Okay, thanks. Yeah.
Thank you so much again, Naveed. I was wondering if others had questions before we bring this to a close. We're a little bit over time. I think we've all learned quite a lot. I think I've certainly, again, learned that there are some things that we know less about than we thought we did. So that's very interesting, but it's, it does show that there's a real way forward and with stroke, which is fantastic to see. And I think, as engineers in universities, enabling together groups like yours and others who are developing these systems and trying to develop standards and platforms, that enable people to use different products like yours, I think would be a fantastic thing. And then we'll be creating that market as fast as possible and helping people as soon as we can. So I'd like to thank you again for your time and the talk, and thanks to everybody who took the time out to join today.
Well, thank you very much. It was a pleasure. You have my email address on the chat, so if anyone wants to reach out, please feel free. And thank you for the discussion and the question.
Fantastic. Thank
Thank
you.
you.
Take care. Bye.
Bye.
Bye.